Welcome to the Chalkless class. This lesson is about the formation of ectoderm, mesoderm, and endoderm, and fetal development. The formation of primary germ layers is gastrulation. First, let's understand the cellular basis of gastrulation, which includes the migratory movements of the cells. The coordinated movements shown by the cells during the gastrulation are called morphogenetic movements. In the human embryo, the cells do two types of movements, epiboly and emboly. Epiboly is the spreading of a sheet of cells on the surface of the embryo. Epiblast cells move toward and into the primitive streak in epiboly movements. In emboly type of movements, the epiblast cells move into the interior of the embryo through the primitive streak. The epiblast cells move into the interior and displace the existing hypoblast cells. Such type of movement is also known as ingression. In addition to epiboly and emboly, human embryonic cells exhibit two more movements, namely convergence and extension. Gastrulation begins with the appearance of a primitive streak. At the beginning of the third week, ectodermal cells proliferate and move toward the midline to form a primitive streak. At the cranial end of the primitive streak, the cells form an elevation called the primitive node. A groove formed in a primitive streak is a primitive groove. The cells of epiblast invaginate through primitive streak to form definitive endoderm and intraembryonic mesoderm. At the cephalic end, a precordial plate appears. It contributes to the oropharyngeal membrane. The oropharyngeal membrane forms the anterior blind end of the gut. It ruptures and forms the mouth. The epiblast cells present on either side of the primitive streak, roll into the primitive groove. They occupy the place present between epiblast and hypoblast. The first collection of rolled-in epiblast cells invades the hypoblast and displaces its cells. As a result of that, the hypoblast was completely replaced by a new layer of cells, the definitive endoderm. The endoderm gives rise to the lining of the future alimentary canal. The migration of cells towards the hypoblast through the primitive streak is called ingression. After the formation of definitive endoderm, some epiblast cells migrate into the space between epiblast and endoderm to form a third germ layer, the intraembryonic mesoderm. The migrated mesodermal cells reorganize to form four main subdivisions of intraembryonic mesoderm. Those are cardiogenic mesoderm, paraxial mesoderm, intermediate mesoderm, and lateral plate mesoderm. In addition to these, a fifth population of mesodermal cells migrate from the primitive node and form a midline tube called the notochordal process. Paraxial mesoderm forms the somites in trunk region. The intermediate mesoderm, also called the nephrotome, forms excretory organs. Lateral plate mesoderm splits and forms coelom. Once the formation of endoderm and mesoderm is complete, epiblast stops the ingression into the embryonic disc. The remaining epiblast cells form the ectoderm. With the formation of three germ layers, ectoderm, endoderm, and mesoderm, the process of gastrulation completes. The three germinal layers arise from the epiblast.
Embryonic endoderm, embryonic mesoderm, embryonic ectoderm, extraembryonic ectoderm, extraembryonic mesoderm, amnion, yolk sac cavity, amniotic cavity, primitive streak.
The precursor mesodermal cells from the cephalic end of the primitive streak move into an embryonic disc and form the notochord. Notochord is a solid structure, forms dorsal to the gut. The formation of the notochord is called notogenesis. The process of formation of the neural tube is called neurulation. About 16th day of the embryo, above the newly formed notochord, ectodermal cells thicken to form a neural plate. Elevated folds of neural plate fuse and form a cylindrical neural tube. The neural crest originates from cells located along the lateral margins of the neural plate. The neural crest produces various structures of the embryo, sometimes called the fourth germ layer of the body. The paraxial mesoderm divides into somatomeres in the craniocephalic axis. After the seventh somatomere, rest of the somatomeres form cubical structures called somites in the future trunk. These are the precursor cells population that gives rise to various structures in the development of vertebrates. At the end of the fifth week, about 42 to 44 pairs of somites are formed in the human embryo. Each somite is divided into three parts: outer dermatome, middle myotome, inner sclerotome. Dermatome forms the dermis of the skin. Myotome forms skeletal muscles. Sclerotome forms vertebrae and ribs. Ectodermal derivatives are epidermis, exoskeleton, sebaceous, and sweat glands. Epithelium of lower part of the anal canal, oral cavity, nasal cavity. Enamel of teeth. Stomadeum, proctodeum. Nervous system. The lens of the eye. Retina. The cornea. It forms internal ear and external ear. Pineal and pituitary glands. Forms adrenal medulla. Mesodermal derivatives are dermis of the skin. Epithelia of body cavity, lymph vessels, blood vessels, and the cavities present in the joints. Skeletal, smooth, cardiac muscles. Bones, cartilage, connective tissue proper, and blood cells. Heart, blood vessels, and lymph vessels. Choroid and sclerotic coats of the eye. Kidneys, ureter. Testes and ovaries. Adrenal cortex. Endodermal derivatives are gut and other visceral organs. Epithelial lining of the gastrointestinal tract. Lungs, trachea, and bronchi. Vestigial gills. Tongue. Liver and pancreas. Thyroid gland and parathyroid glands. Eustachian tube and middle ear cavity. Urinary bladder. Uterus. The upper part of the vagina. Thymus gland. After first month of pregnancy. Formation of the heart. Heart sounds are audible. At second month. Limbs and digits appear. At the end of the third month. Major organ systems develop, such as genital organs. Fourth month from fertilization, face to look human. Cerebral lobes differentiate. The nose, ears, and eyes look more normal. At fifth month, fetal movements, hair on the head appear. End of sixth month, the body is covered by fine hair, lanugo. Eyelids separate and eyelashes are formed. At the end of 9 months of pregnancy, fetus is fully developed and ready for delivery. 266 days from conception, childbirth occurs. Gastrulation at a glance. Zygote undergoes cleavages and form trophoblast and inner cell mass of the blastocyst. Trophoblast further divides into cellular cytotrophoblast and outer syncytiotrophoblast. The inner cell mass of the blastocyst, also called embryoblast or embryonic disc. On the lower side of embryonic disc, a layer, hypoblast, differentiates. The rest of the embryoblast is called epiblast. From hypoblast extraembryonic endoderm and yolk sac endoderm develop. Amniotic ectoderm and embryonic epiblast form from epiblast. On the upper surface of the embryonic epiblast, the cells show migratory movements and form a primitive streak. Morphogenetic movements of the cells through primitive streak result in the formation of embryonic endoderm, embryonic mesoderm, allantoic endoderm, extraembryonic mesoderm, and notochord. From embryonic ectoderm, nervous system and neural crest are produced. Thank you for visiting the Chocolates class.